Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing NEST exam question paper conducted in the year 2024. I will be discussing the biology section. This is session 2 of the discussion. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of NEST being discussed, you can go to my channel Omni Bioentrance. In the channel playlist, see for NEST exam and there you will find. Then next one. First one. A researcher was trying to amplify DNA fragment in vitro using a polymerase chain reaction. All the required components for the reaction except the Taq DNA polymerase were available. The experimenter decided to use DNA polymerase isolated from E. coli instead of Taq DNA polymerase. Okay. The E. coli polymerase was added to the reaction at the extension step of each cycle of the PCR. In this scenario, from the options given below, the optimal temperature to be set in the extension step of the PCR is. So first and foremost, why we use Taq DNA polymerase in PCR? Because it is thermostable. It can have an optimum temperature range between 75 to 80 degrees Celsius. We do not use E. coli DNA polymerase in most of the PCR reaction because it is not thermostable. Its optimum temperature is around 37 degrees Celsius. Now coming to the steps in PCR. You have 3 steps, that is denaturation at 94 to 95 degrees, annealing at 50 to 56 degrees and extension at 72 degrees Celsius. So here what he has done is the E. coli polymerase was added to the reaction at the extension step of each cycle of the PCR. That is around 72 degrees. That is the extension step in when you are using Taq DNA polymerase. In this scenario, that is in case of E. coli polymerase, the optimal temperature to be set in the extension step of the PCR will be 37 degrees Celsius, because at 72 degrees Celsius, E. coli DNA polymerase will denature and it will not function. But at 37 degrees Celsius, which is the optimum temperature for E. coli DNA polymerase, it will function. So the answer here is option B, that is 37 degrees Celsius. The correct statement with respect to arteries, veins and capillaries. Valves are absent in arteries but present in veins and capillaries. Valves are not present in capillaries, so A is not the right answer. Pulse can be felt in veins and capillaries but not in arteries. But that is also not correct, because pulse can be felt in arteries but not in veins and capillaries. So that's not correct. Valves are absent in arteries and capillaries but present in veins. That's correct. Pulse can be felt in arteries and veins but not in capillaries. Again wrong, because pulse can be felt in arteries but not in veins and capillaries. So D is also not correct. Then next one. In the given pedigree, okay, circle represent female, square represent male, shaded portion represent affected individual. Based on the pedigree analysis, consider the statement 1 to 4. Okay. So this is the pedigree, these are the statements, and this is what I have done, analysis. Okay, the inheritance pattern is autosomal recessive. Okay, so here parents are unaffected, if you see here, but offsprings are affected. Here also parents are unaffected but offsprings are affected. So it is a clear case of autosomal recessive condition. If it was autosomal dominant, as they have told in the 4th statement, then there will be no such skipping in generation. That is, parents is not having the disease but the offspring is having the disease. There will not be skip in the generation. All the generation, the dominant disease condition will be shown. So this is not a case of autosomal dominance. So that marks 4th as the incorrect statement. Now read the other statements. The inheritance pattern is autosomal recessive, yes, and both 1, 2 and 3, 8. 1, 2 and 3, 8, that is this one, are carriers. Okay. Now 1, 1 is an autosomal recessive, so naturally it is homozygous recessive genotype. So it will be A, and 1, 2 will be recessive because here the offspring is affected. So it will be heterozygous. Sorry, it will be heterozygous, not recessive. Okay, I told by mistake as recessive, it will be heterozygous. So here this is the genotype of the affected individual. So 2, 4 will be this. So that's correct. Then coming to 3, 8. 3, 8 are carriers of the disease. Yes. So if 3, 8 is a carrier, then only you, and 3, 7 is also a carrier, then only you will get affected offsprings. So these are the genotypes of 3, 7 and 3, 8, and you got affected offsprings. See here. So 1 is correct. The inheritance pattern is autosomal recessive, and the individual 2, 6. Now which is 2, 6? 2, 6 could be homozygous for the normal allele or heterozygous for the diseased allele. Okay. Now 2, 5 could be heterozygous. Okay. 2, 5. The offspring of these two individuals is going to be heterozygous. See here 1, 1, 1, 2 offsprings will be like this. So it is going to be heterozygous. It is not diseased, so it is heterozygous. And 2, 6 could be homozygous for the normal allele. So we will put it as capital A, capital A. So these are the offspring. So you will get individuals that are unaffected. Okay, here upon crossing this male and female, you got individuals, offsprings who are unaffected. So that could be correct. Or it can be heterozygous for the diseased allele. Now if 2, 6 is heterozygous, you will get maximum number of offsprings which are unaffected, and only 1 will be affected. So that is correct. They have not shown the affected individual here. So 2 is also correct. The inheritance pattern is autosomal recessive, and the individual 2, 2 is not a carrier. 2, 2. It has to be a carrier, because only if it is a carrier you will get these individuals as affected individuals. So third one is not correct. So answer here is A, 1 and 2. Now next one. In a plant, allele capital P small p govern leaf color, purple and green, whereas capital C small c they govern the shape of the leaf edge, sharp or rounded. A cross was performed with the two parent plants and the following progeny was obtained. Based on the information, the parental genotypes are. So this is the offspring. These are the number of progeny. Based on the information, the parental genotypes are. Now cross each parent and see if you can get all these offsprings. So these two upon crossing, these are the gametes. You will get some as purple colored with sharp edge. Yes, you have that offspring here. Again purple colored with sharp edge, purple colored with sharp edge. Then purple colored with round edge. Here is another that offspring. So purple colored with round edge is there. Then green colored with sharp edge, this one. Yes, that is there in the offspring here. Green colored with sharp edge again. This is again green colored with sharp edge, and this is green colored with rounded edge. So green colored with rounded edge. So all these four offsprings are present upon crossing these two parents. Now if you see the case of B, you will get only one type of offspring. Similarly, if you cross C with C option parents, you will get only one type of offspring. And similarly D, when you cross, you get only two type of offspring, not four. So the right answer is option A. Then the amino acid sequence of an unknown peptide can be determined using a series of proteolytic digestions. An 8-mer peptide contains 1 alanine, 2 arginines, 1 methionine, 1 serine, 1 tyrosine, 2 valine with alanine as the N terminus, remember that, and valine as the C terminus. This peptide was digested with trypsin. Okay, trypsin will cleave the peptide bond C terminus to a lysine or arginine. That is how trypsin cleaves. And chymotrypsin cleaves the peptide bond N terminus of an aromatic amino acid in two separate reactions. So trypsin cleaves C terminus of lysine and arginine. Chymotrypsin cleaves N terminus of an aromatic amino acid. In these amino acids, you can see that only one is aromatic, that is tyrosine. All the others are not aromatic. So the amino acid composition of each of the fragment obtained below. So this is trypsin digestion fragment, this is chymotrypsin digestion fragment, the correct amino acid sequence. Now such kind of questions you have to practice a lot. Only then you will be able to do it. Okay. Okay. Now see the answer. So this is trypsin digestion fragment. This is chymotrypsin digestion fragment. So they have told in the question, trypsin cleaves the C terminus of the lysine or arginine residue. So here in the question there is no lysine amino acid. So I have written only arginine there for easy understanding. So this is the arginine amino acid. This is the N terminus. This is the C terminus. It will cleave at the C terminus, means it will cut here. Okay. So this will be one half and the other half. Similarly, chymotrypsin cleaves at the N terminus of the aromatic amino acid. So this is tyrosine. Tyrosine is the only one which is aromatic in the given question. So this is the N terminus of tyrosine. It will cleave there. Now now they have also told that alanine is the N terminus and valine is the C terminus. Remember that. In all the options also alanine is the N terminus, valine is the C terminus. Okay. Now trypsin digestion fragment, serine, alanine, arginine. So if arginine is present, arginine has to be present at the third position in this sequence. Alanine is there. So alanine is naturally going to be the N terminus and serine will come in between. So alanine, serine, arginine will be the sequence there. Now there is valine also and fragment 3 which is tyrosine, arginine, methionine, valine. So if you see chymotrypsin digestion, the fragment 2 that is valine, tyrosine, arginine, methionine is the same as the fragment 3 in tyrosine. So from here you can tell that tyrosine will be the beginning because C terminus cleaves the N terminal of tyrosine residue. So it is cleaving here. Okay. So tyrosine will come in the beginning in this sequence, followed by arginine, methionine and valine. And since valine is coming at the C terminus, valine will be at C terminus. So valine I put here, tyrosine I put here, then methionine and arginine I write randomly. Okay. Now you got the first sequence, that is alanine, serine, arginine, that I put it here, alanine, serine, arginine. Now there is a second valine that has to be placed. Tyrosine, methionine, arginine, valine I have put in the end because that is the same sequence that you are getting in chymotrypsin digestion as well as trypsin digestion. So that will be your last fragment. Now there is a valine here. Now from where did that valine come? If you see this chymotrypsin digestion fragment, valine, arginine, alanine, serine. So your first fragment of trypsin digestion, that is alanine, serine, arginine, there is a valine also. Okay, since trypsin digestion, serine, alanine, arginine, it digests at the end terminus of arginine, it will cut here. Okay, but it doesn't, so after 3 fragments there is a cut. After 3 amino acid sequence there is a cut, then comes valine. Okay, that is why chymotrypsin digestion gave 4 amino acid sequences. So alanine, serine, arginine, valine, then tyrosine, arginine, methionine and valine. So that will be your sequence. Alanine, serine, arginine, valine, tyrosine, methionine, arginine, valine. So that will be the sequence. Hope you understood. You can understand this only after practicing a lot. Okay. So alanine, serine, arginine, valine, tyrosine, methionine, arginine, valine. So if you, that will be the sequence. So if you digest this with trypsin, it will cut here arginine and this arginine, and you will get a independent valine as well as a fragment having alanine, serine, arginine and valine, tyrosine, methionine, arginine. Now if you digest the same with chymotrypsin, you will get, it will cut here. Okay, it will cut here and you will get a fragment with alanine, serine, arginine, valine and a next fragment with tyrosine, methionine, arginine, valine. So A is the right answer. The next one. The central dogma of molecular biology states that information flow starts from genome. Consider the following statements. So central dogma, you all know that DNA is transcribed to RNA, is translated to proteins. Then the DNA undergoes replication, producing its own kind. So the central dogma of molecular biology states that information flow starts from the genome. Consider the following statement. Mutation in the DNA dependent RNA polymerase, that is the RNA polymerase that is transcribing your, that is converting DNA to RNA, reverses direction of the information flow. Mutation is not going to reverse the direction of the information flow. It will affect the process of transcription. Okay. So first one is not correct. There is amplification of the coded information from DNA to protein. Now what is this amplification of the coded information? That means one gene DNA sequence can produce multiple mRNA molecules upon transcription. One DNA molecule can produce multiple mRNA molecules upon transcription. And from one mRNA molecule, it can produce multiple proteins upon translation. That is amplification of the coded information. So that is correct. So 2 is a correct statement. The central dogma explains the flow of information in all known forms of life. That's also correct. The genome size determines the direction of the information flow. No. The genome size does not determine the direction of the information flow. So that makes 4 wrong. So your answer is B, that is 2 and 3. Then following are the statements about sun leaves and shade leaves. So sun leaves, they are more exposed to sunlight, and shade leaves, they are less exposed to sunlight. So to increase photosynthesis, shade leaves will have their own mechanism. Okay, so we will see. Chloroplasts are mostly restricted to palisade mesophyll cells in sun leaves. Yes, that is correct. but are evenly present in spongy and palisade mesophyll cells in shade leaves. Shade leaves, they are less exposed to sunlight, so in order to capture more light energy, they have chloroplast in their spongy as well as palisade tissue, mesophyll cells. Whereas in sun leaves, they are always exposed to high intensity sunlight, so restrict them to mesophyll cells on the palisade, that is enough. They can happily undergo photosynthesis more efficiently than shade leaves. So 1 is correct. The density of stomata is low in the sun leaves. No. Sun leaves have higher density of stomata because they undergo transpiration more, and because of the presence of stomata, it will facilitate gas exchange and help in cooling, and high in shade leaf. So statement 2 is not correct. Starch accumulation in chloroplast of sun leaves is higher compared to that in shade leaves. Yes, sun leaves are more exposed to light, so more active photosynthetically. So starch accumulation is more in the chloroplast of sun leaves than in shade leaves. So that is correct. Sun leaves are larger and thinner. No, sun leaves are smaller and thicker, while shade leaves are thinner and larger. This is to optimize light capture. So 4th is not correct. So 1 and 3, that makes B option as the right answer. Then next one. An experimenter attempting to measure the biodiversity of a region has made several measurements in different sampling areas of the region and plotted those values in a graph. The biodiversity of a region can be correctly represented by plotting. So the number of different species versus different sampling areas. Yes. That will give the biodiversity of a region. So you can estimate the total number of species in the region when you plot the graph, and you can compare biodiversity between different areas, as well as you can identify the species richness and distribution also. Number of individuals of a species versus different sampling areas, that will give only data regarding species abundance and not biodiversity. Number of primary producers in the region, again it will show productivity, not biodiversity. Ratio of number of land species to aquatic species is not sufficient. You have to go for the number of different species itself. So D is also not correct. So A is the answer. Ribosomal rRNA sequence analysis can be used for the identification of bacteria. An experimenter was analyzing total cell lysate, that is homogenized mixture of a plant tissue that is heavily infected with bacterial pathogen. The lysate was used to prepare ribosome enriched fraction with large and small subunits dissociated. So the experimenter, he took the plant tissue which was infected with bacteria, isolated the ribosome enriched fraction and this ribosome enriched fraction has large and small subunits, that is large subunit and small subunit of the ribosome dissociated, and this was subjected to density gradient centrifugation. The resulting banding pattern is shown in the figure. The correct set of bands that the researcher can use for the pathogen identification, that is bacterial identification. Okay, so this is the tube showing density gradient centrifugation. So the bacteria, you know, the small subunit is 30S and large subunit is 50S. And plant, the small subunit is 40S and large subunit is 60S. So upon density gradient centrifugation, they will separate as follows. So this band at the top will be bacteria 30S ribosome, that is the smallest, least dense, so 30S ribosome. The second will be plant 40S ribosome, this one. After 30, the next highest is 40, so that will be 40S. Then the next highest will be 50S, so it will be 50S of bacteria. and the next will be 60s of the 60s of the plant. So here they have asked for pathogen identification, that is for bacterial identification. So the subunits will be 30s and 50s. They come in 1 and 3. So band 1 and 3 are that of the bacteria. The other 2 are of the plant. So 1 and 3 is the answer. Then accumulation of H2O2 is harmful for the survival of eukaryotic cell. The cell level of H2O2 in the cell is kept under control by. So it is by peroxisomes, because they have the enzyme catalase, and the catalase, it breaks down H2O2 into water and oxygen. So the answer here is option C.